Plastic is everywhere in our daily lives, from food containers, cars, electronics, medical supplies, you name it. But all this convenience comes with a major downside. Every year, about 8 million pounds of plastic end up in our oceans. In 2015 alone, the world produced more than 448 million tons of plastic, and that amount is expected to double by 2050. Once plastic gets into the environment, it breaks down extremely slowly and releases tiny pieces called microplastics, along with chemicals that can harm wildlife and even our health. These chemicals are linked to issues like cancer, hormone disruption, reproductive problems, and heart disease. On top of that, making plastic in the first place produces huge amounts of greenhouse gases and requires clearing land that normally absorbs carbon dioxide.
In 2017, something surprising happened that gave scientists a new idea. Frederica Bertocchini, a scientist and beekeeper, found that waxworms in her hives, which were normally considered pests, chewed right through a plastic bag in under an hour. Waxworms are the caterpillar-like stage of the wax moth. They hatch from eggs, spend most of their life eating things inside beehives, like wax and pollen, then form a cocoon and eventually become moths. The adult moths don't eat at all. They're just focused on laying eggs to continue the cycle. Because the larvae feed on materials inside of hives, beekeepers often see them as pests, though they're a popular food source for reptiles, birds, and fish.
After Bertocchini's accidental discovery, researchers took a closer look and found that waxworm saliva contains special substances called enzymes that can break down one of the world's most common plastics, polyethylene, the stuff used in grocery bags and many food containers. There are 4 enzymes in the saliva of waxworms: Ceres, Demetra, Cibeles, and Cora. These enzymes can start breaking plastic apart in just a few hours at room temperature, a process that normally takes decades in nature. Even better, some of these enzymes can help transform old plastic into material that's as good as new, opening the door to true recycling where plastic doesn't lose quality each time it gets reused. The Cora enzyme forms a pocket that interacts with a free tryptophan, which is one of the many building blocks for proteins. The actual function of this pocket is unknown, but it seems to help with the breakdown of plastic. In our research, we wanted to learn more about one of these enzymes called Cora and whether wild waxworms and commercially raised waxworms have differences in the gene that produces it. This matters because understanding how the enzyme works could help scientists recreate it in the lab, meaning we might someday break down plastic without needing the worms themselves.
To investigate this, we collected waxworms from different places, extracted DNA from their heads, and focused on 2 regions of the Cora gene. We copied these regions by performing PCR, a method of repeatedly copying a specific section of DNA, which makes it easier to study. In our case, we replicated exon 3 and exon 5 of the Cora gene. An exon is a section of DNA that codes for a protein. We then did gel electrophoresis to confirm that our PCR worked, a technique that uses electricity to move segments of DNA through a gel. We used this method to make sure we only had DNA from the exons we wanted. After this, we cleaned up our samples and had the DNA sequenced so we could read their genetic code. We used BLAST, a government database of DNA sequences, to confirm that we had the right segments. Finally, we aligned and compared the sequences for the exons in wild and commercial worms.
What we found was that the Cora gene is almost identical across all the worms we studied. There were a few tiny differences, single-letter changes in the DNA, that showed up more often in commercial worms, but overall the gene was highly similar. This usually means the gene is important for the worm's survival. When a gene plays an essential role, it tends to stay the same over generations because worms with harmful mutations in that gene don't survive long enough to pass them on.
Even though we've learned a lot, there's still much more to discover. Scientists hope to eventually produce these plastic-breaking enzymes without needing the worms at all. We also still don't fully understand why one part of the enzyme binds to a certain molecule, or how all of the enzymes in waxworm saliva work together. By continuing to study these details, we may one day develop powerful tools to help reduce plastic waste and protect the planet.